Hello, I'm Thomas Chiozzi. I'm a software engineer at DNA Algo, and today we will talk about the encoding and decoding pipeline of data into synthetic DNA. What you see here is a generalized overview of the steps that are common to many encoding strategies. Of course, this is intended as a loose guideline. A particular or specific encoding solution might differ from the proposed approach. As you can see in the scheme, the minimal steps necessary to encode binary data into DNA are the application of an error correction code, the division of data into smaller chunks, the application of an index to each chunk, the mapping from the binary domain to the DNA domain, and the assignment of a primer to each DNA sequence. Now we will slightly focus on the role of each step in the encoding pipeline. First, an error correction code is used. An error correction code will append extra data to our information so that the information can be recovered even when the data is slightly corrupted. In this slide, the extra information is highlighted in blue. Then the binary data is split into smaller chunks. This is because you can only write small DNA sequences, and when dividing data into smaller chunks, you start to create the information that will eventually become each individual oligonucleotide. Then, to each chunk, you attach and evaluate a unique index. The role of the index will soon be clear. Afterward, to each chunk, you perform a translation from the binary domain to the DNA domain. Finally, to each oligonucleotide, you attach a primer. As you can see from the picture, the primer is common to all sequences and will uniquely identify the binary file. The reason that we use an index is that when reading DNA sequences, the data might be shuffled around. So in order to recover the original ordering, you must use an index and evaluate the index. The mapping will do mostly what it says. It translates information from the binary domain to the HGT domain. You can see in this table a very simple form of mapping that translates 2 bits into 1 nucleotide. But note that some technologies are not able to write arbitrary sequences. Some sequences of DNA might be challenging, and you can fit those requirements to the mapping strategies that can be adjusted accordingly. What you see here is the decoding pipeline. Decoding will take noisy DNA information that were resulted from the sequencing machine and will recover the original file. The decoding steps are mostly the encoding step done in reverse order. So first, we remove the primers. Then we invert the mapping and go from the DNA domain to the binary domain. Then we use the index in order to merge the information in correct order. And finally, we use our error correction code to decode the information and try to correct some form of corruption that resulted in noisy DNA sequences. If everything went smoothly, you will have a recovered file that is equal to the original file. You might see that in the slide there is one extra step that is called clustering and tracer extraction. A DNA sequencing machine will read any given DNA sequence multiple times, and each observation might have unique errors. Here at DNA Algo, in order to maximize the quality of information passed to the decoder, we aggregate similar sequences according to the metric of edit distance. In this slide, you will see this task with the name of clustering. Then, from the aggregated data, we try to obtain a higher quality consensus that will be decoded. Sometimes this task of obtaining a higher quality consensus is called trace reconstruction. We have observed what are the minimal steps necessary to encode and decode information onto DNA. Thank you very much. Hope you enjoyed it.